Good evening. I'd like to welcome everybody to the Iowa State University Extension Dairy Team virtual chat number 5 for dairy and poultry producers. Back when this ailment first appeared, we started doing these chats, and as the title implies, this will be the 5th one. Dr. Philip Jordan and I will be moderating this evening and helping with the questions. For voice recognition, I'm going to ask each of our presenters to say hello. Dr. Pat Gordon, you'll be the first presenter. If you'll just kind of let people know what your voice sounds like.
Good evening, everyone. Thanks for joining.
Dr. Sato.
Hello, this is Yuko. Thank you for coming.
Dr. Kazan.
Good evening, everyone.
And Jürgen Eiler.
Good evening, everyone. Jürgen Eiler, Iowa Department of Agriculture.
Thank you very much. We'll go ahead and each presenter will have some time to talk about what they're dealing with, and then our questions will come at the end of the presentations. So, if you have questions you want to make sure get answered, if you want to type them into the chat box as the program's proceeding, that will kind of expedite getting things out there that we can organize. So, Dr. Jordan.
Okay, I'm going to kick things off a little bit, just a little bit of background and the outbreak. As everyone knows, this started happening in early this year, 2024. First, it was in Texas, New Mexico, and Kansas, but unfortunately, it spread to the rest of the country, some other states. What we saw was a decrease. The big major clinical signs were decrease in feed intake, decrease in milk production, and then an off-colored milk. And then it was diagnosed as high-path avian influenza H5N1 and reported on March 26th. But those states in the South United States have been dealing with that for a couple of months. And then since then, it seems to have decreased, but we still have a big outbreak going in California right now. I want to just give a little background in the virus. As we talk, we're going to talk some things about the testing and what it means. So here's a picture of the virus, and that H5N1. What that means are these hemagglutinin things that are in this model here is related with this blue color, and in neuraminidase this red color, and there's a whole bunch of different ones of these. This particular one right now is H5N1. But as you can guess, if you multiply 11 times 18, there's lots of different types of avian influenza A, and the varying amounts of pathogenicity amongst them. And then inside here, there's these things that are represented here with these green curly cues. Those are the ribonucleic protein complexes. And when we get to some of the testing, that's actually what we test for, both in the when we do the checks for the RNA in it, or for checking for the antibodies. But since this is an RNA virus, that's one of the reasons it mutates very quickly, because RNA viruses don't have the mechanism to correct themselves, so they mutate quickly. And then if a cell gets infected with two different types of this virus, they can exchange these strands with each other. So there's two ways that it mutates quickly like that. And this is the pumpkin that Dr. Sato and I made for the pumpkin contest, and we won first place for the scariest pumpkin. Then I want to show this graph here of people have started calling this the Magstead curve. Drew Magstead's one of our clinicians or pathologists here at Iowa State. This was a report from a dairy in Texas early on in the disease, and you can see here this dairy was going along with just less than 8 cases, eight new sick cows a day. Then all of a sudden here, where they got this black line drawn, something bad happened on this farm. There's a bunch of sick cows. They went from having less than 8 a day up to 30, 40, 50, up to 60 a day. And at the same time, the somatic cell count went up some. So that's the background of that dairy. And then this dairy, as Dr. Megstead said, hey, we have some samples in the freezer from before this outbreak happened. Would you like those? And he said, yes, I'd like those. So he took those and did a test on them called a PCR test, which is a polymerase chain reaction. And the way we measure that is with the CT threshold. So this is CT stands for cycle threshold. It's essentially the number of times that this test has to repeat itself and to get enough virus to be found. So it's really confusing because the lower the number means that the more virus was there to start with. So as was showed it here, we have up here we have 64 is the above that we don't check it. So about 2 weeks before, about 17 days before we had this outbreak, you could start picking up a little bit of this virus. And then at the time that it was the peak, there was a lot of virus in there. So this gives us a little bit of confidence that this herd, it's a new disease. It wasn't something that had been there before and we just missed it. Unfortunately, in dairies, these dairies seem to clear it. Of course, we don't know that for 100% sure, but after about 3 weeks after this happened in this particular dairy, we could no longer find the virus. And then we could also do another test called the ELISA. It's a test for the antibodies, and it too is a little bit confusing. We measure it with this S to N ratio, and that also a lower number is actually more positive in this particular ELISA test. In some ELISA tests it's not, but in this one that's the case. So here at the beginning, they were negative up here at 0.9. And then about the time we started being showing clinical signs, it went down fairly quickly. So we look at this, we can actually tell if a herd's infected by doing the PCR test. And we can tell if the herd has been infected using the ELISA test. And then just a little bit about the phylogenetic tree here and how that works. This particular virus got this really long name. It's called high-path avian influenza H5N1 because those are the things that are on it. And then we have these further definitions, clade 2.3.4.4b, and then genotype B3.13. So these are all just clarifications. So this is kind of like saying this is a Angus cow, and these are types of relations to it. So it's fortunately for this particular outbreak, we've not seen a lot of changes in this. It's still B313 in the dairy herds that we're seeing. So that's all I have. I'll take questions later and turn it over to Dr. Gordon now. I'll stop sharing.
Hey,
So can,
Phil, before you go away, can you put your slides back up?
yes.
Can you go back to that slide with the number of cases and the CT values? Back one. There you go.
This one?
Yeah. Well,
Oh, no.
This one here.
Okay.
I just wanted to point out that the CT values are actually on the right y-axis, not the 64 down to 0.
Oh, sorry. Yep.
That was the number of cases. So
Yep. Very good.
if anybody
Thank
thinks it's running a CT up to 64, we're not.
Okay. Yeah. Sorry. Okay. So next speaker is Dr. Gordon. He's one of the clinicians at Iowa State. He's also on a lot of committees that have been working with this virus through ABP and the National Conference on Interstate Milk Shippers and other groups that have been meeting regularly. So he's been involved in a lot of the discussions from the start on this. So go ahead, Dr. Gordon.
Thank you, Dr. Jardin. Again, good evening, everyone. As Dr. Jardin indicated, I have been involved as a liaison to the National Conference of Interstate Milk Shipments for just a bit over 10 years. and have been on their executive board for several years of the last over the last several years. As the conversations began to happen about developing a national strategy to control the disease and to test milk, I was asked to participate in a team within the organization that developed this new program. And Dr. Jardin already showed these two slides and indicated that primarily the disease is out in California and a little bit of Nevada right now as far as the dairy industry goes. So the national strategy to get to the point where we can say that the dairy herd is free of H5N1 is displayed here. Currently, we're setting up in area number 1 where we're going to begin this what we call a national milk plant silo monitoring program, which will allow for us to determine individual state status, whether it could be non-detected or non-negative. And then eventually we will get over to the point where the goal is that we have freedom of H5N1 in the US dairy population. So to get there, we are this week embarking on a strategy where individual states, their milk regulatory individuals like Jurgen's group here in Iowa are collaborating with the USDA and traveling out to milk plants in their states and collecting samples from milk silos, storage silos within the processing plants. Those samples will be sent to the National Veterinary Services Lab here in Ames, Iowa, out along Interstate 35, and will be tested via PCR for the presence of the virus in the milk samples. Now those milk samples will be from potentially from multiple dairy farms, most likely from multiple dairy farms. And so the milk regulatory people will also collect from the plant the bulk tank units, which are an identifier of regions of dairy farms that are kind of similar in nature, that are those being assigned by the individual processors. So in Iowa, there are some bulk tank units that maybe only have one dairy farm in them, and there is some that have over 100 dairy farms in them. So there's going to be quite a bit of work to be done should a silo plant test positive and it has a bulk tank unit in it that has over 100 individual dairy farms making up that bulk tank unit. Because once they have determined that a initial sample is non-negative, they will work with the state veterinarian and the USDA people within the individual states to go out and collect samples from each of the dairy farms that would have gone into that individual silo and then test that milk likely at the diagnostic lab associated with the state or near the state through the what we call the National Animal Health monitoring system, or the laboratory monitoring system, sorry, NALAN system, which is a cooperation between the state diagnostic labs and the USDA. So if a farm a silo is found to be positive, it'll be reported back to the individual state, and that state will become what's called an affected state. So right now, as an example, we have active disease in California. If a silo would be tested in California, it would very likely come back, or there'd be a high likelihood that it could come back and would be considered an affected farm, an affected state. So California likely will kind of start in this affected state area. If after 4 rounds of testing, and the idea is that milk samples will be collected approximately once a month, and then no more frequently than 4 times every 6 months. So approximately 4 to 6 months down the road, if a state has gone through a series of testing 4 times, we will be to the unaffected state status. So what we'll start to see as you go to the USDA's website is a map similar to what I showed you a bit earlier that will show the affected, unaffected status of the individual states. And then as we develop control programs in the affected states to try to control the disease, hopefully we will move more and more states into this affected stage 4, the unaffected state status, and then eventually move the entire country into a stage 5 situation where all surveillance and monitoring and testing is showing up negative over time. USDA thinks that this is probably a 2-year process to get to stage 5. But that will really depend upon the aggressiveness of the testing and controlling infected animals from moving from farm to farm, because that's one of the major risk factors for moving this virus around the country. So with that, that's kind of the program in a nutshell. Jürgen and Dr. Kaizen probably have more comments on it in a bit. So I'm going to turn it over to our next speaker.
All right, so our next speaker is Dr. Yuko Sato. She's our extension poultry veterinarian at Iowa State and one of the pathologists at the diagnostic lab.
I'm going to be pretty brief with my section. Oops. So some of the common questions we're getting is like, seems like now, so for those of you in the audience that's not from Iowa, we started to see HPAI avian influenza cases in Iowa. And the general vibe is like, this seems like this is a sudden surge of events. Is this something new? Well, not really. So if you look at it, it's a seasonal event. If you look at the migratory waterfowl pathways, we're kind of in this Mississippi flyway, central flyway, kind of the congregation of here over here. And if you look at basically data over time, if you look at where the birds migrate, you see peaks during the springtime, and then you see a peak during like the fall, winter time. So this is essentially in accordance with what we're seeing over here. If you guys haven't had the chance to look at a website called Birdcast, it's a website that basically tells you how many birds are in flight and what direction are they flying. So I just looked up what we saw for yesterday, and it kind of tells you how many birds are in flight at a given time. So this was yesterday at 9:30 p.m. And then you can see there's 9.6 million birds in flight. And you can see the direction of the arrow. Again, makes sense. Birds are migrating from north to south, kind of snowbirding over here. And some of our first cases have been detected kind of in the northwestern corner of our state. And you can kind of see that heat map showing where the birds are migrating. And the question is, okay, is this related to the dairy HPAI strain? The short answer is no. The strains we're seeing right now in the commercial poultry industry cases have been all migratory wild bird or wild bird strain origin influenza. But can this happen again? That's a big question. When we approached some of the scientists about like how this monster of a virus has been created, first of all, like Dr. Jardin kind of set the stage by saying like B313 is the kind of the name of the genotype of the virus. It's still an avian virus. It's not a cattle virus. It's still an avian influenza, but this is basically a recipe for disaster for an opportunistic event to happen. So in order for this to happen, you have to have multiple animal species and potentially near parturition that are present. you have to have ducks and some other poultry present in the facility. And those animals share a common water source. That seems to be what may be the recipe for disaster of what happened with B313. So again, even though what we're seeing now in the commercial industry is a wild bird strain of the virus, can this happen again? You know, I don't have a magic 8-ball, but it's a freak event, and how many freak events can happen? Who knows? So that's all I have, and I will pass it on to the next speaker.
Okay, so our next speaker doesn't have slides. He's going to talk. Is Dr. Jeff Kaisan, who is the state veterinarian in Iowa.
Thank you, Dr. Jardin. And IDOLS greatly appreciates the opportunity tonight to give you a little information from the department, where we're at, actually what's going on today with HPAI and some of our next steps going forward here. Dr. Gordon and Dr. Sato have done a great job of giving an overview of both what's rolled out from the USDA here recently, in conjunction with the federal order and silo testing, the phased approach to moving towards a nationwide elimination status. Dr. Sato's given a great job of giving an overview of HPAI and poultry. I'll go into a little bit more detail here in a minute on our current status in the state of Iowa. The first thing, though, I'd like to do is maybe take a step back where we kind of left off at the beginning of summer. Our cases of HPAI and dairy cattle included 13 premises in Iowa. We also had 3 poultry facilities that were infected with the B313 strain. And as we've indicated before on these chats, our approach at that time is, and one of our main responsibilities, or one of the biggest responsibilities, is our job within the Department of Agriculture, IDOLS, is to protect the health status of all animals in the state of Iowa. And as we've discussed, this situation has been a very difficult situation where we have one species where we take a completely different process than the other. Where one species we completely depopulate a premise where the birds are infected, require the producer to clean up the site before they can restock. Whereas in dairy, we did not, and there's no state that's taken that, where the dairy cattle are depopulated. It has varied a little bit from state to state on the approach with it, with specific dairies, whether quarantines are issued or not issued. And so as a review of what we did in the state of Iowa earlier this year is we did not quarantine affected dairies with this B313 strain. We did work directly with those dairies to make sure they were taking as many precautions as they could on their own in regards to biocontainment, stopping the spread of disease from their farms to any other farms. We encouraged them to make sure as they were moving cow cows off the farms, that they weren't moving wet cows, if at all possible. If they were, that those went directly to slaughter. So we did not take an approach where we quarantined individual dairies infected with this strain. When we found that the 3 poultry facilities had broke with the B313 strain, we did start a strategy that fit right in with our normal strategy of what we do around poultry premises. Is we establish control areas and require farms in that control area to test. And in those control areas, normally what we do around infected poultry premise, we require the other poultry premises to test. And when we set up a control area, it's because there are farms near an infected farm and they're determined to be at higher risk because they are near an infected farm. So we also started an approach. We were the first state to required all dairies in that control zone to test as well. And we did, as everyone should remember, we did pick up some dairies that were positive by that testing. We also had a couple dairies that were outside of control areas that reported clinical signs. One of the other things we did earlier this year is we changed our reporting rule. In rules, we require we required previously that for high path, that all poultry was required to report any clinical signs of high path. We switched that. We did an emergency rule that stated that all species have to report any clinical signs that could be associated with high path. So we made reporting high path required in all species. Now the good thing, since early summer, early June, we have not had any other cases in dairy. So we're hopeful as we move forward, as we'll discuss here shortly, the testing that's going to happen moving forward, because we've not had any reports of clinical signs in dairies. And what was also just previously mentioned of the status around the United States, that there are not many states reporting active cases besides California and just recently Nevada. So we've not had any reported cases in dairy or poultry until just very recently. We in the last, and this is a point where I wanted to make sure that I thank a couple of groups. I want to make sure everybody understands we have a staff that works very hard within the Department of Agriculture and IDALS, not only with the animal industry division, but support from other divisions. And in the last just short of 2 weeks, that group has been working very hard on what I'll describe in a minute, what's going on in high path. So I want to make sure everyone realizes that there's a tremendous amount of work that's being done behind the scenes, and there's a great support staff that we have within IDALS. The other piece, we need to give a big thank you to the poultry industry, and specifically the poultry producers that have been affected just recently. They've done a tremendous job on depopulation, staying current, because one of the things that we did find from 2015 and that we've practiced through this outbreak since 2022 is rapid depopulation of poultry facilities greatly enhances the ability, or we have shown that it stops the spread from farms. That all of the introductions for the most part since 2022 have been lateral introductions into dairy farms. So again, as I've mentioned, just short of in the last 2 weeks, we've had 8 premises infected with high path avian influenza. One of those is a backyard flock. There's 4 layers that have been infected, and the total inventory of those 4 layers together is 6.6 million birds. We've also had 3 commercial turkey facilities, and between those 3 sites, it's roughly 106,000 birds. And I can tell you, as of today, even finishing up today, all of those birds have been depopulated, and disposal is well underway in those sites. Most of the way those animals are being disposed of is composting. There is some burial being done. We are watching very closely the sequences of those farms. And as Dr. Sato mentioned, so far, all indications are that this is not the dairy strain affecting these poultry facilities. It's the migratory birds moving through, and what we're seeing in some other states. One of the strains is a D1.1. There's some other reports of D1.2, but as of right now, it is not shown to be the dairy strain. We are watching that very closely, and if that were to switch, we will quickly move back to where we stopped early in the summer, where if we have a poultry facility infected with the dairy strain, we'll institute again control area testing of the dairies in that area. But again, as of right now, that's not the case that we've found, but we're watching very closely. So I think I will stop there. Hopefully I've given at least a brief snapshot of where we're at today, where we've been, where we would pick up today if we were to start experiencing the dairy strain again in poultry facilities. So I'll stop here and turn it over to Mr.
All right, so our next speaker is Jürgen Ehlers, and Jürgen is the Bureau Chief of the Dairy Products Control Bureau. And Jürgen, maybe you can explain a little bit more about what that job exactly is.
Sure, absolutely. So my ultimate responsibility is the sanitation surveillance in accordance with FDA and USDA's uh, protocols. And so I am in charge of all of the licensing and permitting of farms, processing plants, haulers, anything involved with the movement of milk up to the distribution. So from farm to distribution, those responsibilities fall to me and my staff. Thank you to everyone already tonight that's presented. It's made my job really easy. My responsibility here will be to quickly just tell you where we are at for the state of Iowa in terms of the plant silo monitoring process. Dr. Gordon already has showed you the phases of that process. And so I will just real briefly tell you where we're at with that. The intention for sampling is only revolving grade A milk intended for pasteurization. And so in that process, we have identified 25 facilities in Iowa that meet that requirement. In other words, they are receiving in and or redistributing or pasteurizing milk intended for pasteurization. The goal within our department is to sample approximately 110 samples per month. And Dr. Gordon alluded to the 4 out of 6 sampling process that is in accordance with the standards that already are present in the pasteurized milk ordinance. And so the goal was to allow my staff to already continue what would be routine monthly regulatory sampling. And so we will be just adding this component onto it. What will happen is at each of these sites indicated, we will collect samples. Those samples will be submitted to the NBSL lab. It is important to know that the only information that will be recorded on the submission forms is the bulk tank unit associated with the silo. So any milk that goes into that silo or storage tank, storage vessel, the bulk tank unit, which Dr. Gordon alluded to, is either a farm or a combination of farms. The number associated with those farms will be recorded on the submission form. So if there is a identification of positivity, that will be reported to the state veterinarian, Dr. Kaizand, and then it will become our responsibility to, based on that BTU number, determine the farms that are associated with that silo sample. So that at that point, we would be in conjunction with the facilities to identify exactly what producers went into that particular silo. The good news is we have already started that process. We are one of 13 states that is participating in some extent of this 5-phase program. That information was came out today. And like I said, Iowa has already started the process. So today and yesterday, during routine monthly sampling, my inspection staff has already collected those samples, and those samples are on their way to NBSL for testing. Also, as Dr. Gordon alluded, there are secondary steps in the phase out program, which at some point, either with positive identification of producers or in phase 2, which is essentially testing all producers, we will be able to determine if and where the virus is located in Iowa. So I think at this point, that's about all I have to share.
Okay. So if there's any questions, people can come off mute or put them in the chat. I'll start with one for whoever wants to answer it. What exactly is test going to test for? Is it going to test for the virus or the antibodies or both?
Yeah, I can grab that one. So the initial testing of the plant of the sample that is submitted to NBSL will be a PCR test, similar to what Dr. Jardin described there a bit earlier. If the sample is found to be non-negative, USDA will also attempt to do an NP ELISA on that sample. And then also, if there's enough material available, they will also try to sequence the virus to know what the genotype of the particular viral material that's being found in the sample, to figure that genotype out. They will not be doing any virus isolation of those samples at this time.
All right, thank you. All right, another question here. What's the timeline for getting results? And will the milk results be shared publicly? Like what positive tests have been in the past?
That's still a bit being worked out. NVSL has stated that they intend to be able to turn sample results around in approximately 5 days. I think that may include the shipping time also, but I'm not 100% certain on that. The team that I'm working with through NCIMS will meet tomorrow to start to look at how the data is going to be displayed on the website, but it will be on the same USDA website where the high path data is shared for both the poultry and the dairy. So I believe there'll be a map available to start to show the results, but I'm not sure of the timeline, whether we'll see any of that data in January or if it's going to be a bit longer before it's officially available.
All right. Any more questions?
Hi, I have a quick question. So did I hear correctly that all of the testing will happen here in Ames at the USDA facility across the country? All the samples will come to Ames?
Yes, that's correct. All the samples that are collected by the milk regulatory people of milk silos will initially come to NVSL at Ames.
Okay.
The follow-up testing should a silo test positive via PCR, the follow-up testing that would be done to determine the farm or farms of origin would be done through the local diagnostic labs within the NALA network. So those are labs that have been certified by USDA to perform the PCR and associated testing.
Great, thank you.
I do see a note here from Dr. Beckett Weddle that that poultry reports county level, dairy reports state level. And she says results for milk will only be reported at the state level once state vet notified. Is that correct?
That is correct. The processing plants will not receive the information back as far as whether or not they had a silo that was positive. It will be only going to it will only be going to the state veterinarian. I think it's really important to point out and continue to make people aware of the fact that this is an animal health issue. It is not a food safety for humans consuming the product issue. There has been multiple tests, multiple surveys that have been done on pasteurized product, and that has repeatedly demonstrated that while you may be able to find portions of the virus in pasteurized milk, no viable virus has ever been found at any samples that have been have been tested. So pasteurization is very effective of our milk products. And so that's the reason to continue to work towards this is an animal health control program.
So she says only testing grade A milk destined for pasteurization.
Only testing grade A milk destined for pasteurization. That is correct. So any milk that would be initially or eventually going towards a raw milk like cheese product would not be incorporated into this testing this time.
But they clarified that on the talk last week that if there's grade B and grade A mixed together, then they will be tested, right?
That's correct.
If it's destined for pasteurization. Yeah.
Right.
So it's good. It's going to be most of the milk in the United States. It'll be probably 98%.
I think it'll be closer to 99%.
Okay.
And it only includes cow milk. It does not include other cloven hoof mammals.
Right.
Okay.
Because we've got folks from out of the state of Iowa, with the comments you just made, the media's picked up quite a bit that there was been raw milk called back because of HPAI found in it. So is there difference in states? Are they testing differently?
Fred, you're talking about the incident in California, correct?
Correct.
Okay.
Actually, I think it's 2 incidents now, 2 different raw milk dairies.
Right. I'm only aware of one. So I think it's important to clarify a difference here. The milk program that is run underneath the pasteurized milk ordinance is milk that is intended, for the most part, to be pasteurized as part of the process before it becomes a food product that's offered for sale at the grocery store or wherever you may pick it up. The situation where what that Fred is referring to was actually milk that is actually intended to be consumed raw. So it's a slightly different intention. They both use the word raw in their in the definition, but in this program, all this milk is intended to be pasteurized. We don't have a good idea about the danger of consuming raw milk that would be infected with this virus at this time.
So out of curiosity, do you know how they identified the raw milk in the California situation, the virus in the raw milk? Were they routinely testing that, or do you have any idea? I'm just curious. Oh, and Dr. Bickett-Weddle says California has been testing raw milk dairy since May. So thank you. That that does clear that up. She asks if there's any testing of raw milk dairies in Iowa.
For H5N1, I assume.
Yeah.
Yeah. So in Iowa, we do have producers that fit a certain description have the ability to sell raw milk in the state of Iowa. That is not milk going into interstate shipment program like that we're talking about here tonight. Within the guidance provided from the legislature in this raw milk program, it does indicate that a veterinarian is to perform a health test 1 time a year to certify that the animals are healthy. As a veterinarian who consults with raw milk dairies in the state, or veterinarians who work with raw milk dairies in the state, I do recommend that they're testing their milk for H5N1. I think it's an important enough pathogen. There has been some human disease associated with dairy farm workers and poultry farm workers that I think it's a test that should be considered for producers who are producing raw milk for consumption as raw milk.
Pat, whether it's goats or sheep or dairy cows, raw milk, isn't it required in that same legislation that they require a monthly milk test or a periodic milk test?
Yeah, so the raw milk legislation requires a period or a monthly bacteria count, both the total bacteria and then also what's called coliform bacteria, which are essentially the E. coli type bacteria that are found in fecal material, as an example. That will not pick up this virus. It's a different organism, and the type of testing that we do to enumerate the bacteria that are present in the milk will not tell the producer whether or not there's a virus present in the milk or not.
If the producer has a concern, can they request that on their monthly sample?
Yes, absolutely. They should work with their veterinarian and find out the proper sample and how to collect it. Realistically, if they were sending a milk sample to the veterinary diagnostic lab at Iowa State to have the bacteria counts done, they could use that same milk sample after the bacteria count testing was set up to be able to confirm, or subset it off to be able to do the PCR testing to confirm the presence or absence of H5N1.
We have a question also from Jennifer asking, after we get to the country to stage 5, will there be further testing? I see smiles, but no.
I think that's a question for the state animal health official.
So I guess the answer is there's a fair number of these details being worked out yet behind some of these phases. So we do not know that what the requirements would be as if we were to get to a eradicated state.
If folks have a question, they can put it into the chat box. We've had several comments and questions there, or they can unmute and ask the question directly. We'll give just a minute or so for folks to formulate those questions. And then we'll ask for some closing statements or closing recommendations to our milk producers and poultry producers. Dr. Sato, I didn't want to be one-sided there, so.
I do appreciate Danelle's comment that viruses don't read bills and don't read legislation. So we did not anticipate until March that viruses of public health concern would be in cow's milk. So I did chuckle at that.
Yeah, I give a lecture in Gail's class to the senior animal science students, and I gave it in 2023 and went through all the things that cause mastitis, and there were bacteria and yeast and fungi and everything like that in there, but no viruses. So I had to add that this year.
I guess that's job security for the veterinarians and researchers on the call.
Well, let's go down the list of the order that they presented. Dr. Gordon, last comments?
No, well, I should say yes. Thank you for the opportunity to present. It's hopefully this will be one of the last times we have to do this, but I doubt it. I think if you're managing cows or advising producers on managing cows, it's really important to realize that the biosecurity, and Danelle is going to plot even louder as I say this, is the most important thing that we can do to control the spread of this virus from an infected farm to uninfected farms. That is repeatedly being shown in the epidemiological surveys that have been done, that moving infected cattle from site to site has been one of the major causes of how this virus is spread. So just be diligent on your biosecurity and cattle movement practices. Thank you again.
Yeah, I'll make a point there. When this first started happening in March, in some of these early meetings we had, we talked a lot about bird control, thinking this was if it came north, it was going to come north on birds. But it's been pretty well shown that we've brought it north with dairy cattle movement, not with birds. Sorry to interrupt.
Dr. Sato?
I'll make a comment. This is less related to the dairy side, but again, the virus we're seeing right now in Iowa is related to a wild bird strain. And one of the first ways we know what kind of virus is out there to figure out that link is to find dead wild birds in the area. And that kind of gives us an idea of basically what's in your neighborhood. So I had, I think in another, I guess, news media report, Dr. Rachel Rudin, who's our DNR veterinarian, mentioned about reporting if you find wild birds in the area that are dead, don't touch them. Call your local DNR staff. I'll put a link in the chat and call that person, let them know that they're in the area. They can go collect the samples, and that could be our first indication of what's to come, essentially, so that producers can ramp up their biosecurity. So that was kind of a plug on the poultry side. Appreciate the opportunity to join our chat today.
Dr. Kazan?
Sure. So again, IDALS greatly appreciates the opportunity tonight to present and go over some of these things, some of our next steps, and we're hopeful that we've not had any reported cases in dairy that we've moved through with this silo testing, and it's uneventful. But depending on what we find in that silo testing will kind of determine our next steps that we take at the state level. Probably to re-emphasize what's been said and we've said before on some of these chats, and I know it's easier said than it's done on the farm, but control your own destiny. It's been mentioned biosecurity here, but the first thing producers, whether they're poultry or dairy, is make sure biosecurity is in place on the farms to protect from everything that you can put protections against on your farm. We're still learning a lot about this, on how it's spread, whether it's poultry or dairy, but there are some things, even has been mentioned here, there are some things that can be controlled on the farm. So as we've said for years, control your destiny on your farm, make sure you're protecting what you can.
And Jürgen Eiler?
Yes, just thank you all for your cooperation, working to combat this virus. We really cannot do it without everyone, and we appreciate forums like this, sharing the voice so that we're all on the same page. So thank you again for allowing me to participate.
I'd like to thank all of our presenters. They brought forward a lot of good current information, and I'll kind of harken back to one of the earlier virtual chats. Dr. Sato made the comment that this works best when we communicate, whether it's a poultry producer, dairy producer, an academic, a researcher, or the veterinary that we work with. So I think those are great words of wisdom then, and I think they're reinforced with the comments we heard this evening. So, Dr. Jordan, have you got any closing comments?
Nope, I think that's a very good summary.
Very good. We thank everybody who attended, and as Dr. Gordon said, hopefully we won't have to do this again, but we don't have that crystal ball yet. So thank you all for attending, and we appreciate your time.